Hi, everybody. Thank you for attending our second Bair Ranch seminar of the semester. Our speaker today is Dr. Sally Johnson. She is the Paul Mellon Distinguished Professor of Ag at Virginia Tech, and she's also the current editor-in-chief of the Journal of Animal Science. And so we're very thankful that she's traveled from Virginia to be with us today. I also want to take a moment to acknowledge Treston from the Bair Ranch Foundation. We'd like to thank you and the Bair Ranch Foundation for the continued support for our department, as well as the seminar series. And with that, I will turn it over to Dr. Johnson.
Thank you. I don't have my glasses.
I'd offer you mine, but I don't think they'd do you any good.
Well, thank you for coming out to hear today's seminar. It's a real treat for me to give this seminar because historically, this is not where I usually spend most of my talks. So I thought I'd kind of give you the brief roadmap, if you will, for how I kind of went down this twisted road to actually end up working with horses at the moment. So I am a Michigan native, not the University of Michigan, but Michigan State. So I grew up on a farm. My family has a very small beef cow-calf farm. Nothing like it is here. I think my dad has 100 cows. But I grew up there. I enjoyed that part of agriculture. I did not enjoy the concept of vet medicine. But I went to Michigan State because that's where my family goes. We all go to Michigan State. And so I stayed there. I have an animal sciences degree from that university, and I stayed for my master's degree as well. And during my master's, I worked with a very eclectic person called Werner Bergen. And Werner is kind of the pioneer of growth biology. And he introduced me to skeletal muscle and skeletal muscle growth. So my master's project was one of the pioneering studies where we proved that ractopamine needs 7 days to actually improve protein synthesis in the pig. So a 7-day project is always kind of fun for a master's student. Working with pigs convinced me that I didn't enjoy that part. And so I moved on, and I went as far away as I could possibly get. So I moved to Arizona. And I worked there with Ron Allen, and Ron Allen is, I guess in the animal sciences world, would be known as the father of satellite cell biology. And I learned from him so many things about this incredible population of stem cells. And I've always maintained my interest in satellite cell biology since my PhD degree there. I took my first job at Penn State, and I stayed with satellite cell biology there. The lab group that I led exploited some of the differences in signal intensity and signal strength from an intercellular perspective and how that controls cell fate. I really enjoyed my time there. My husband was recruited to Florida, and so I joined him. And so we moved to Florida, and I took all of the concepts that I had developed at Penn State with me to Florida. But I had to change my model. So I went from working primarily with chick embryos to Florida, which is the land of eared cattle. And I got involved with the differences between Angus and Brangus and Brahman from a growth perspective. And so the satellite cell work that we were doing there kind of transitioned into that. And from that, we started to recognize that you could really do a lot controlling cell identity by changing the differences in growth factor, as well as the signals that they would use when responding to those growth factors. And it was there that we decided that you could take a unipotent satellite cell and convince it to become more ES-like or embryonic-like. In order to have a robust cell population to say, well, if we can make an embryonic-like cell, can we actually make it convert all the way back? And so the logical cell type for us to do that with was going to be a cord blood cell. I did not want to work with the hospital and going through the process of IRB. So I'm kind of anti-legislation in general. So we decided to work with the horse because in Florida, It's a huge place to foal out mares. And we worked with a barn that foaled 150 mares every year, and they would give us 50 mils of newborn foal blood every year from that 150. And we were able to discover that using human reagents, you can isolate cord blood stem cells from that horse. And so we have tanks full of these, should the future racehorse ever want them. So that's kind of where I got started. I went from chickens to cattle to horses, but I've always maintained my interest in satellite cells. So what are these things? So satellite cells have been around conceptually for a very long time. So in the 60s, an electrophysiologist, an MD, by the name of Alexander Moreau, was working with frogs and frog muscle in particular, and he was using electrical stimulation to study contraction. And he discovered a set of cells that was located on the outside of the muscle fiber in a satellite position. And he noticed that with a strong enough contraction, that these cells would actually reawaken. So normally they are very chromatin dense in a quiescent state. And he thought, wow, this is really interesting. So maybe this population of cells is the responsible cell for giving rise and solving what he called the vexing problem of skeletal muscle regeneration. So remember that we all take lots of beatings in our muscle. We always take lots of hits, and we always repair ourselves. And it was the same thing in these frogs, and as well as other species. So when you have this huge myotrauma, you're able to repair yourself. So is it these cells that was actually contributing to that or responsible? Because at those point in time, a lot of people thought that if you damage the muscle fiber, and again, the muscle fiber is a very long cell with thousands of nuclei in it. And the thought was, if you damage it, then those myonuclei contained within that fiber could reassemble and become proliferative and form a new fiber. Kind of the blastoma theory behind some of the wound and tail repair or regeneration that you would see in tadpoles. So from that, we had our initial discussion and discovery and definition of what a satellite cell was. Since that point in time, and that was in the 60s, so you can transition through until probably the mid-90s. There were very few papers actually published on satellite cells, with most of them very descriptive, add and subtract proteins and media and see what happens. But in about 2000, I think it was, Mike Runicki was doing a subtractive hybridization screen between proliferating satellite cell isolates and those that were fresh. And through this molecular technique, he identified a gene that was highly enriched in this quiescent population. And by sequencing, it was identified as PAX7. And so he went on and showed that PAX7 marks all of the satellite cells that you would see in muscle. Okay. And so that was our first big breakthrough, because now we have a way of actually identifying the cells so we can follow them and ask, what do they do and how are they controlled? So from that, we now know, there. We now know that these PAX7 cells are going to be referred to as the myogenic stem and progenitor population. Okay, so satellite cells suddenly got a really fancy definition as stem and progenitors. And by this definition, it's also apparent that satellite cells are a heterogeneous population. So it's not one specific cell type. It's actually a stem cell and its offspring that are referred to as satellite cells. And these cells are going to be entirely responsible for regeneration. So in mice, if you isolate these PAX-positive satellite cells, you'll have a lot of mononucleated cells. They grow, and then they're going to begin to express something called myogenin. And this is a transcription factor you have to have in order to make a muscle fiber. So in vitro, you'll begin to see this multinucleated fiber formation. And then ultimately, these fibers are going to express myosin, as a contractile protein. If you take mice and you genetically ablate PAX7, and you do that across the genome, those animals are born, they usually are going to die within 5 days, okay? And the reason those pups die is because they don't have a diaphragm to support breathing, one of those essential features of life. And so the diaphragm is very thin, and with that respiration movement, it actually ruptures and they will suffocate, basically. So we knew that happens. Of those that were hets, you could see some defects in muscle repair regeneration as well. And so what they've done since that point in time is to create conditional mutants. And so if you conditionally knock out PAX7 in an animal that is alive and well, you will actually disrupt all of regeneration. And so that, again, tells us that you have to have these cells in order to repair your muscle. Since that point in time, we've gone on and we've tried to assign their function, if you will, to the process of hypertrophy, because that's always been kind of a conundrum for muscle biologists. And we know that based on the mouse models that you use, that yes, the satellite cell is responsible for some of that cross-sectional area increase in size and muscle growth that is responsible for muscle formation. So how does this actually occur? The satellite cell is an adult stem cell, and you are born with these cells and you will die with these cells. So they're a stem, and it means that they continually are going to be able to repopulate themselves. It's one of the primary features of a stem cell. And they do that by both symmetric and asymmetric division, which we were talking about earlier today. And so most of what we know about this comes from mouse models, okay? In the mouse, you can label the satellite cell and you can watch it divide using time-lapse videography. And what you'll find is that the mother satellite cell will divide, will retain the DNA, the parent DNA in herself, and the newly synthesized DNA goes on to the offspring, which is the progenitor cell. And that asymmetry is what allows us to continually have a stem cell pool. It's not to say that the stem cell can only divide asymmetrically, okay? It is a preferred way, but they can also divide symmetrically. And so using, again, time-lapse video in the mouse, and these are helping mice, what they can find, at least in the tibialis anterior muscle, is that a stem cell in a young animal will divide symmetrically once, and then will go on to divide asymmetrically 3 times. And the ultimate outcome of that is going to be about 48 progenitor cells. And so the beauty behind all of that is when you actually look at transplantation experiments, because if you take one mother cell and put it into a muscle bed that has been damaged, she can completely repopulate that entire muscle. So if you take out the entire tibialis muscle of a mouse and you replace it with 6 satellite cells, a new muscle will form from that. So there's a huge amount of regenerative potential that is retained within these satellite cells. And that has a lot of beauty for us as biologists, as well as therapeutically. Because in skeletal muscle, one of the things that historically has always been popular for us is how do we cure muscular dystrophy? And so based on the regenerative potential of these cells, if we can harness it and come up with creative ways to introduce these cells into young men, we could possibly repair some of their loss of muscle. So that's nice when we're talking about disease and damage. But most of us are healthy individuals, and our livestock are healthy, our horses are healthy. And so what is the importance of satellite cells for normal function, and what is their importance for exercise? And so that's where an area that has become very popular these days is what is the contribution of a satellite cell to both repair post-exercise, as well as what does that new addition of the cell to the muscle mean for what we call muscle memory. So in this case, most of what we have available now comes from mice and humans, okay? And using these two model systems, and the model here is going to be healthy young men under the age of 30.
The only way you can convince them to let them take a biopsy.
Yes, desperate for cash. And so that's where a lot of our exercise experiments are going to come from. And in people, you primarily are going to be resistance training, or you're going to be doing some endurance training. And we can mimic those sort of for mice with different types of wheel running and different types of tail weighting to mimic those two things. But when we do that, and we look with a single bout of exercise in these two model systems, we look at the satellite cell in tissue sections. Okay, so these young guys, they come in, they do their exercise, and we take a biopsy from the vastus. You cut tissue sections, and you can look under the microscope, and you look at PAX7 positive by immunostaining. And what you'll find is that 24 hours after exercise, most of the cells, satellite cells, PAX7 here, are typically quiescent, okay? 24 hours later, if you undergo resistance exercise, they're going to be sitting in this phase where they're considered activated. Doesn't mean they're dividing. It simply means that they're primed and they're ready to divide. If you are an endurance runner, okay, this is usually a bike model. In this case, you do your single bout of exercise. This PAX positive cell, normally quiescent, 24 hours later is already transitioned through this and is found in this condition. Okay, and so those cells are actively divided. So these would be the progenitor cells that we spoke of earlier, which would be PAX7, usually marked with PAX7 and MyoD. And so they, if you pulse with BRDU or EDU or thymidine analog, you would see them as having some incorporation of EDU, meaning that they've gone back into the cell cycle and have gone through S phase. So exercise has a response within 24 hours in these two model systems, okay? So that says that the cell has detected some kind of a damage response, and they need to go on to repair it. That's what their job is. If you are a trained athlete, okay, you have the same progressive model in response to endurance and resistance. The only thing you have more of is total numbers of satellite cells, okay? So usually you're going to have, in this system, I think it's like 2 to 3 nuclei per 100 satellite cells per 100 fibers, and that'll increase to about 4, okay? So you have more. Satellite cells, as a percent of your total population of a muscle, is 5% of all nuclei. Okay, so there are very few of these, but they're very powerful. So what does this mean for the ultimate athlete? Okay, so if we're going to study exercise, what does this mean with the horse? Instead of get you kind of gain you a perspective of why the horse really kicks man's butt. We can kind of walk through man versus beast here. Okay, so these are Olympic caliber sprinters. That person gets up to 36 kilometers per hour, and he can hold that speed for about 9.4 seconds. Okay, a horse, 64 kilometers per hour, and can hold it for 2 minutes. Okay, before fatigue sets in and he has to stop. So immediately greater speeds and longer times. In order to do that, you think about the muscle and its demands for oxygen, okay? So how is it going to meet that respiration demand? In people, and again, trained athletes, the maximum oxygen consumption is about 85 mils of oxygen per kilogram of body weight per minute, okay? The horse, 180 to 200 mils of oxygen per kilogram of body weight, okay? So if you start doing the math, remember a horse is about 520 kilos, okay? And so this number gets to be huge in a minute. That's how much oxygen it'll consume. So a huge amount. How do you get that there? Well, it's not because your heart rate changes, right? So man and beast are both 220 beats per minute is maximal heart rate, okay? The maximal heart rate doesn't change. Max means max, okay? But what is very different is size of your heart. So I think we all, in high school, I think they tell you, make a fist, that's your heart. So that's about 320 grams in your average Olympic athlete, okay? A horse? No. That's about 1% of body weight. So that's 7 to 10 kilos in some of your elite athletes. Okay, so a huge heart. If you have a bigger heart, you have a bigger left ventricle. If you have a bigger left ventricle, you can pump more blood because that's the pump of the heart. Okay, so the cardiac output, or the amount of blood you can pump per minute in people, is about 20 to 24 liters in a minute. In the horse, about 240 to 450 liters in a minute gets pumped throughout there. So you have a larger heart that pumps more blood, that carries more oxygen to meet the oxygen demand of functioning muscle, okay? And this is really important because for the purposes of this talk, a horse to me is a thoroughbred. Not to be rude, but it's just a thoroughbred. We've bred them, they're 55% of body weight is skeletal muscle, okay? So they're a chunk of meat. All right, that is a lot of muscle. Man, probably 40, maybe you can get 45, maybe. So they have a much larger muscle mass. So their demands are going to be so much greater because they're going so much faster. But they have the system that allows them to do that. And the other point that I'll make about oxygen with the horse, and I think sled dogs as well, they will have their spleen constrict when you hit about 140 beats per minute. Their spleen constricts and you get an increase of 30% red blood cells into the bloodstream. So you can even carry more oxygen to meet the demands of muscle. Huge athlete, beautiful horses to watch. But they get tired. Man gets tired, horse gets tired. And fatigue is a multi-component process, okay? They can't meet the oxygen demands anymore. Lactate is accumulating in the blood, and lactate is actually a marker for a skewing in electrolyte imbalances, and it's a skewing in loss of bicarb potential in the muscles. They lose buffering capacity as well. You have inorganic phosphates that are accumulating in the muscle. The other thing you're going to do is your core body temperature is increasing, okay? And so that's one of the things we always worry about with the horse is because core temperature increases, but the horse's neural detection of fatigue is a mystery because man, there's a mental component to why you stop, okay? And the horse, it's debatable whether the horse has much mental capacity to detect any kind of fatigue. And so the thought is that that's why some of these horses will run till they just drop into the ground, is their ability to have a brain that says stop it is not there. The neural connections are a little different. There's a woman in University of Dublin, Emmeline Hill, who's an equine geneticist, who's identified a SNP for a gene that actually is connected to neural detection of fatigue. And in the thoroughbreds, they think that this may be part of why some horses will just keep going forever. The core temperature is a problem for us, right? Especially when you can't tell yourself to stop. Core temperature, if you hit 42, you're probably going to die. 42C in a horse is probably death. Exhaustion, so how do we know? Because we don't want to hit that point, right? We want to stop. Exhaustion, you start to see an increase in creatine kinase in the blood, myoglobin in the blood. These are things that are normally only found inside of a muscle fiber. So when you see that, it's a kind of a crude indication that the fiber is leaky, has suffered some damage, and so these things are coming into the bloodstream. Again, part of the fatigue identification process. And there's other markers of damage that people will look at as well. Energy repletion. So one of the driving forces behind this fatigue is that you're out of energy. So you don't have enough oxygen to make things happen to begin with. But the other component is in man, your muscle glycogen becomes depleted. So you probably lose 30 to 40% of the muscle glycogen you have stored. You get tired, you stop, you come back, you drink Gatorade. The sugar goes, you replenish your glycogen, you're good to go 24 hours later from an energy repletion standpoint. In the horse, he runs the Derby, he doesn't get back the 30% of the glycogen he lost for 3 days. Okay, so there's a delay in that, which is a delay in metabolism possibly, which can contribute, we believe, to why the satellite cell is a little bit different for the horse. So if we've experienced exhaustion and if we're hurt and we've got to start thinking about getting our satellite cells moving so we can repair ourselves. And there are 3 primary phases that we have to think about here. So satellite cells are going to be located outside of this fiber, distinct from this huge multinucleated muscle fiber. It will see HGF produced both here by the fiber as well as by itself. And HGF is believed to be responsible for activation. Okay, and so that tells the cell to reawaken, come out of your deep quiescence and reawaken. You don't want to come out and not be able to go back, right? That's part of self-renewal of stem cells and maintenance of that population. And to help us with that, we have this group of peptides and proteins, myostatin and TGF-beta. So myostatin, I think most of us think of myostatin as being the gene that's disrupted with double-muscled cattle, okay, Belgian blue and Piedmontese. Myostatin is a brake, and it tells the fiber to stop growing, and it tells the satellite cell to stop dividing. And so that's where it comes in. HGF tells you to come out of quiescence. Myostatin, TGF-beta are thought to put you back into that. And then the last group of proteins that we need to think about are these 2, FGF2 and IGF-1. And both are made by the fiber. FGF2 is probably the most potent mitogen that we know for satellite cells. You can usually get at least a 2-fold increase in cell numbers when you treat with FGF. IGF is considered anabolic. So IGF is thought to promote fusion and is thought to promote protein synthesis, etc. And so more of a muscle building for that. So with those 3 things in our toolkit, then we need to be able to say what happens in the horse with regard to those local factors being made and satellite cell activities. So this is what happens in a horse, okay? And this is a slide that was adapted from the Japanese racing group and who have done a lot of work in this area. So in this case, you work a horse to exhaustion on a treadmill and you take serial biopsies over time, okay? If you do that, and this is just a shameless plug for Silver Charm, because I have a mare by his so he's due to foal soon. So in this case, you work these horses, you bring them back, you take a biopsy. Within an hour after you've worked out, you see a little interleukin-6 coming up, and that is a pro-inflammatory cytokine at this point in time. In mice, actually, interleukin-6 is part of a proliferative phase for satellite cells. Within 1 day, you see IGF-1 coming up, as well as myogenin. And remember, myogenin is the gene that causes cells to fuse, okay? So it tells the satellite cell, move into the fiber, contribute your nuclei to make more transcripts, to make more muscle proteins. So a day later, that's what you see. Remember, in people and mice, if this was one day, whoop there. If this was one day post, this would have a lot of PAX7, because this is when the satellite cell is active in people and mice. The horse, this is what you see. 3 days later, you sort of see a little PAX7, and I would argue it's just because they added an extra horse to the group. It's not very robust. And so in most of the time, at least in our hands, and we use, for all of our studies, we use 24 horses, we have not been able to replicate this. Okay, we have not been able to say that PAX7 is increased by RNA at 3 days. Instead, this is when the satellite cell in the horse becomes active. Okay, 7 days after a workout, and this is a strenuous workout, okay, but work to exhaustion. And so this is important to think about if you are a horse enthusiast, especially racing, because remember, those horses, your high-end horses, probably race 8 times a year. And a lot of it may be simply because they don't even begin to repair after a race until 7 days later. Okay. In fact, if you go to the backside of a track, a lot of the old-time trainers that are there, they'll tell you a horse doesn't even begin to recover for the first day because he won't eat for the first day after he works. Okay, so you can't even get him to replete the glycogen levels because he's not even going to eat. So what is it? Why is this time frame so delayed in the horse compared to people and mice? Okay. And that's where we've kind of spent the last couple of years in my group, because I think this gives us a wonderful extended time frame to ask, what are the regulatory transitions between there and here that you really can't do in people and mice, because 24 hours, you've got all of this, 48 hours, you're already fusing. And at 7 days in a complete necrotic model in the mouse, you've actually got fibers again in 7 days. So it's much too quick. So one of the first things we had to do was ask, well, how do you activate a satellite cell in a horse? Okay. And for us, we do a traditional activation assay. And so we isolate our satellite cells from these beasts. Our isolation protocol gives us about 97% pure satellite cells. So it's very clean preps. If you culture those in vitro in a growth media, you won't see any kind of division until 72 hours. And so this is when you start to see cells that are actually in S phase. So in this particular graph, about 18% have gone through S phase of the cell cycle. So they've left G0, gone through G1, and now are into their S. 96 hours, everybody is proliferating. Okay, so they're up and rolling. So we can use this assay to ask questions about activation, because if something activates, then this number is going to increase here. Okay, and most likely you'll start to see it happen in here. So we looked at all of these growth factors because they were expressed earlier in that damaged muscle in the horse. And to make a long story short, we were disappointed because none of them, including HGF, which is absolutely required in the mouse. If you don't have HGF, you don't activate your cells. Okay. None of these affected the activation period. So they didn't ask the cell to exit G0 any quicker. Okay. FGF2 and HGF are mitogenic, because this being control cells shown here, about 40% are EDU positive. Both of them are significantly above that, indicating mitogenic activity in response to those particular growth factors. We've gone back because we thought, well, nothing ever works well in a vacuum. It's probably not a single factor. So let's repeat everything again and ask, what happens if you do combinations? And again, in that activation assay, with this being the control. The only combination that actually increased activation was FGF, HGF, IGF combination. And remember, all three of those were present at the 7-day period. So possibly you need to have all three of them in order to promote progression from G0 and into the cell cycle again. If we continue to look at these at 96 hours, and again, this would be proliferation phase, what you find is that, sure, it worked here, but it's not what's needed to sustain the proliferation. So once they're out, the identity, at least in this gamish, is somewhat changed. And so now this cell population is going to be receiving information, asking it to do something else. All of these in green are still myogenic, with the exception of IGF and FHI. So the thing that stood out to us was this. When you have IGF, you tended to see a reduction of proliferation. And you saw it mostly in this group. So IGF is a protein that is found here initially within that first day. In the mouse, you look at IGF and you add IGF to any kind of mouse cell culture, myoblast, and the first thing you get is myogenin expression, because myogenin is a target gene for IGF. And so what most people will tell you is that IGF turns on myogenin tells the cell to fuse. Okay. So we asked, is that what's going on here? To make a long story short, no. Okay. So in the horse, again, if you work them to exhaustion, you bring them back to the barn, and you do a 1-hour biopsy, a 1-day, and a 3-day biopsy. And you look at them by histology for central nuclei. If a fiber has a central nuclei, it means you had a satellite cell fuse into it. So it's a crude estimate of satellite cell activity. So at 24 hours, so that's the 1-day period that the Japanese saw with myogenin, you saw a significant increase in the numbers of central nuclei. And so that is saying that satellite cells are active there. And because there is no PAX7 at that time, only myogenin, it says you probably have a population of satellite cells that are lying next to the fiber, engaged and ready to fuse. So the damage that a horse sees and experiences can begin to repair as soon as 24 hours, based on that subpopulation. So then we asked, okay, what's IGF's role in this? Because, there's all kinds of cheaters out there. And growth hormone is a wonderful cheater drug. And growth hormone causes IGF to increase. And so if IGF is truly involved in this, you would expect to see myogenin increase and more central nuclei. And what we find, at least in vitro, is that's probably not going to be the case. If you treat those cells with IGF-1, what you find is that here, no increase in myogenin, okay? So this is the control cells, which are in a permissive media, where you have the opportunity to divide, or you have the opportunity to go and become terminally differentiated and express myogenin. So in that permissive media, about 10% are going to begin to express myogenin here. If you add IGF, it does nothing. And we've gone back and we know that IGF does not improve fusion rates. It does not do anything to proliferation. And it's active, okay? So it's not because we're adding junk to our culture. So we're not sure what goes on with that, but what we do think is that at least the horse has multiple subsets of satellite cells. So with that in mind, we kind of know what we're going to do with the cell. We want to improve its activity so we can cut back on repair times and maybe get an extra race in. And this is an effective thing to do because of the high stakes that are involved here. So what we've done is we've tried to look at diet and workload. and how could we use these two things to improve repair recovery rates. And we had some preliminary data where you can feed tributyrin to horses. You can work them out, and you actually see an increase in the percentage of active satellite cells. So at least tributyrin, if you supplement that, that's one way to improve the repair process in the horse. We have recently finished a project looking at HMB, and HMB is commonly used in people, okay? HMB is a leucine metabolite. So it's naturally occurring in the body, which allows you to supplement it. And so in this case, what we do is we gear up horses when we have a lot of stuff we do to them. These are expensive studies, so we try to get as much as we can out of them. We'll attach EMGs or electrodes to 3 different muscles in the body. They all wear heart rate monitors, and then they go down to the barn, they work on a treadmill. Okay, and it's a standard treadmill test it usually takes about 15 minutes to reach exhaustive fatigue. And we will capture, you can see here is a little viewing station. So all of the runs are captured on high-speed motion capture cameras. So we analyze all aspects of the biomechanics, as well as capturing electrophysiology. And then they come back, and we also can look at blood biochemistry and muscle histology. So what did we see when we fed this? When we fed it for 6 weeks. In the horse, we like to feed diets a minimum of 6 weeks because myosin has a half-life of 30 days. And so if you go for a month, you're probably just starting to see it. If you go 6 weeks, you're confident. If we do histology for the 3 primary fiber types in the horse, which are type 1, it's a slow twitch oxidative fiber. Type 2A, which is fast twitch oxidative, and then type 2X, which is fast oxidative glycolytic. In our horses, what we found is that HMB actually increased the percentage of type 2A fibers. And so you gained faster fibers that used more oxygen. And this is important because when you put a horse in a training program, they train to gain more type 2As. So by doing this, the thought is, wow, you can kind of shorten that training period a little because you're going to gain 2As this way. And then you just begin to train them to gain the twitch and contraction speed and the fitness that they're going to need. And they gain these at the expense of 2Xs. So you lose some of these 2X fibers. And loss is a relative term because you go from 2X back to 2A. When you're done with it, you take them off the feed, the 2A fibers will transition back to a 2X. Okay. So if you have more 2As, does that mean you're a better athlete? No. And this is probably the PI's fault on this one because these are unfit horses. Okay. And so unfit horses are just unfit. And if you give them more 2A fibers, it doesn't translate into better performance. They still have got to undergo training to improve the cardiovascular fitness that allows them possibly to experience fatigue resistance. Okay, so a 2A fiber is more fatigue resistant than 2Xs. And we saw nothing there. These horses exhausted after about 12 minutes in both of the SETs. If you look at lactate, and the lactate goes up because oxygen becomes limiting, so pyruvate is not taken to the mitochondria for TCA cycle. It's actually in the absence of oxygen goes to lactate and is pumped out of the cell. And so lactate goes up, and there's no difference at 2 minutes, which is the end. And there's no difference in this at 20 minutes after racing or at 60. So the recovery period is still the same. And glycogen loss is still the same in both of them. So it's really not a glycogen sparing event either, with one about 35%, the other one about 32%. So nothing really big there. If we had probably had them in a training protocol, we may have been able to see something different. And we based that on the fact that we looked at some EMG activity. EMG is in some ways, all it does is it tells you more about your action potentials in your muscle. Okay, so contraction gives you an idea of neuromuscular contraction. So as you would expect, these horses wear an EMG here, here, and then on the sides as well. So biceps femoris is the one that we primarily pay attention to, as well as the semitendinosus, which is in the rear. We look at these activity scans, and so the electrodes capture it. This is the raw feed. This is just an example. And so this would be a contraction event, contraction all the way through. We filter it out to get rid of some of the noise. We rectify, which means we only look at the top part. And then we smoothen it out so that we can see some spacing in between. Again, all computer stuff. And then for analyses, what we do is we take this area as the root mean square. Okay, and so that's the data that we'll show you next. So in this case, IMG or electrophysiology, the action potential was unaffected by the diet. Okay, so as you would expect, in this case, this is the right ST and electrical activity in microvolts per second. So with increasing speed, increasing contractions, because you got to get your butt moving, you got to take more steps or you fall off the back of your treadmill. So you get an increase in that microvolts per second. Okay. Fatiguing threshold, and this is a math equation basically, and what it is you linear regress it, and it comes back onto the y-axis in meters per second, and it tells you what the horse when the horse started to experience some fatigue. And so both of these started to experience fatigue at about 10 meters per second. But he continued, remember, until some of them continued out to about 13 meters per second. Okay. So at 10 meters per second, the muscle was actually starting to tire. And 10 meters per second is a full gallop. So we really didn't see much there until we went to this last slide, which is fiber recruitment. And in this case, we look at median frequency. Okay, and so in this case, what we found is that the voltage, if you take the amplitudes on the IEMG signaling, the electrode signaling, and median power frequency is, in this case, at what point in hertz is that bisected evenly, okay? And with fatigue, you see that shift, okay? Because you can't recruit as much. And so the frequency is going to increase. And so in this case, what we're seeing is that the ST, there was a diet effect, okay? So in this case, the median frequency was higher in this case, so meaning that it had shifted so that it was at a further into the rightward shift of that IEMG signal, meaning that it was more fatigue resistant, okay, but had a greater capacity to resist fatigue with that diet. But that didn't translate again into any athletic performance. They all died at the same time and speeds. Biceps femoris, again, some similar things were happening there as well. And so we think that HMB increases median power frequency in the biceps, and that's probably related to that fiber type shift. And when you shift to the 2A, you become more fatigue resistant. And so we're seeing that, but because the horses are unfit, we couldn't really test this diet and prove fatiguing resistance because of that. It became a fitness issue for the major reason why they did not perform. So we have high hopes to repeat a lot of this. I rely heavily on interns. To put them in a training program, we have 24 horses that have to be worked daily. I'll give you Sunday off, but that's a lot of hot walker time. So I only have 2 hot walkers right now, so you got to load it twice anyway. And we only do, again, we live in Virginia. It gets hot, and so you have to get out of bed, you have to be down at the barn, and you have to load the hot walker at 6 o'clock in the morning. That really kills a lot of interns. And so you work them, it's a 1 hour workout, you have to bring them back, you got to wash them out, then you got to feed them, then you got to muck, and then, oh yeah. I welcome interns. So we're going to try and repeat this with fit horses, okay? And so we'll put them in a 6-week training program, and then they'll come back in and they'll do a modified exercise test. So the high-speed exercise test that we did in the past involves go until you can't, okay, with gentle encouragement. And gentle encouragement for us is you bang a whip on the outside of the bar, and they think it's scary, so they keep moving. They don't get whipped. For all of my friends in Iacocca, we don't whip horses. But we do bang a lot, and if you don't like the bang, then we let you off. But we've gone to a different system where we actually work to what we call V200, which is the speed where the horse's heart rate reaches 200 beats per minute. In colloquial terms, this is the equivalent to VO2 max, kind of maximum oxygen consumption. And so we let you go at that speed until you don't want to go. So in conclusion, this I just love, this dope. This is actually Santa Anita. So a few years ago, this guy jumped the fence and decided to run with the horses. Probably not the smartest move he ever made. He was not killed, but he was tackled at the end. So equine muscle recovery is protracted, okay? People, 48 hours, you're probably going to be good to go. And I think we all recognize that, right? We all watch football and we all watch basketball. And, you know, they play every Sunday, okay? So usually pro football players will probably get Monday off, maybe do something light on Tuesday at the most, but Wednesday you get back after it. The horse, you got at least 7 days before you really want to start thinking about how to work him out again. Satellite cell activity occurs in waves during the recovery period. I think this is one of the coolest things ever. So we have a big effort ongoing in this one where we are isolating cells at that pre and post in that 24-hour period. We have contracted with a company called Single-Omics, and they are doing single cell RNA sequencing in those so that we can conclusively identify that we do have a myogenin population of cells there. With the idea is, can we see what else is underlying that myogenin expression? Because it has such therapeutic potential even for people as well. If we could arrest cells at myogenin, you could immediately begin a repair process much earlier. And diet can alter fiber metabolism, which may or may not translate into performance benefits. Okay. I think there's a lot of, of course, people know we sell foo-foo dust by the buckets. And there's all kinds of supplements that are thought to improve performance. And it's always been a big issue, at least in the racing world. I think it was in the last 2 years, a huge number of trainers and veterinarians were arrested by the FBI in a sting operation because they were doping horses. And it's really fun for me. I don't support doping, but what they were giving is a cocktail that included IGF, HGF, and FGF. And so it kind of indirectly added credence to some of the things that we've been trying to explain. So they kind of short-circuited my ICOC and actually got it done. So, but that's where that's kind of our long version of the horse world as far as satellite cells are concerned. I'd be happy to take any questions. Yeah.
You were talking about the time that the satellite cells are becoming active and improved. You've been able to isolate that to the different muscle fiber types, or is that just like within muscle in general? Would that even make a difference?
So yes and no. So you have 2 primary fiber types, fast and slow. A slow fiber has more nuclei, or satellite cells associated with it than your larger type 2 fibers do. And that's been kind of an evolutionary conserved finding. So I believe it was Sarah White found that in the horse, that, yeah, you have more fibers in a type 1 than you do a type 2. And you do that by histology, because you can't isolate individual fibers to retain the cells long enough to examine that.
Fair. So I am kind of thinking selfishly about myself. I'm certainly not a sprinter, but I run marathons. So I would not be a racehorse. I'm thinking like a ranch horse, maybe. So we're not going really fast, but we're going much slower, but for longer distances. Do the satellite cells act differently, or do you know if the timing is different in how they act between those really quick burst athletes versus those long?
Yeah, so in athletes, the resistance or interval training individuals, they are going to have, well, in both cases, you're going to have more satellite cells. If you're trained, you have more than slugs. Of those, if you're a resistance and interval, you will have more at 24 hours that are in that primed period. Okay, so they're activated. And they identify those with PS6 kinase immunostaining, and that cell is considered alert. And so you have that opportunity to go on and repair if needed, or if you're not needed because you're super trained, then you just go back G0. If you are the endurance athlete, and I would argue you're probably more endurance, even though it's anything more than a mile is endurance. So if you are here and you're trained like you are, okay, you at 24 hours after your next marathon are going to be at that proliferative stage, okay? So you actually will repair faster because you've already left G0, gone through activation, and have hit the next phase. So you would repair sooner than an athlete that was like a sprinter or the weightlifters. And you have more satellite cells to begin with because you have more type 1 and 2A fiber. So you would have, just a greater number to help you with.
So we could use her for muscle biopsies.
You hold her down, I got the gun. We'll go lidocaine optional, wrong one. Yeah. Just side it, you'll be fine. So, my definition of a hard workout probably was, I think that's been the conundrum, right? Because this is a conversation we have all the time about that crazy, where they say a hard workout's 90 beats per minute. I know. So resting heart rate for the thoroughbred herd that I maintain is 35. Or closer to 50 because they're and get up to 220. So a hard workout for them is probably not until you hit V200, right? So it has to be at least 10 meters per second for at least, and they'll hold that for a good, that would be more. But workload at V2. That might be sufficient to get you back the next day. Something that's less than, maybe you only get 70%, 45, so that's considered the submax. That for 45 minutes. It's not a hard workout. That's what they do every other day for 6 weeks. Would be 45 minutes, which is 3 sets of walk, trot, canter. 5, 7, 5. 5 trot, or I'm sorry, 5 walk, 7 trot, 5 gallop. Got 3 sets, 45 minutes. Average probably 140. It doesn't do anything. They'll still go out and go around in the paddocks. Yeah, I think defining workload is a huge thing for the lab. A lot of our horses are wearing inertial sensors. And so we all wear Fitbit watches and Apple watches, all that good stuff. So we have the equivalent for horses. And we base it on football patterns and tie it back to their heart rate. And so we can calculate caloric burn. Try to design diets based on how many calories you spent versus how many calories you think your horse spent. So yeah, workload is a big deal.
But you see like an association between being more trained satellite, but is that also like 3 marathons a year to somebody that's trained to do 4 marathons? Satellite cells are quicker recovery.
No, because the training period is. Rodrigo, you're in that. So the training, as soon as you are considered fit and in that training protocol, so usually within that 2 months in people, you're going to have that max satellite cell numbers.
Okay, so that's kind of my question. Is that a max, like the max?
Yeah, and so the really cool stuff that's happening in the world now is, so you train, you have more satellite cells. The satellite cells are actually fusing to repair, but they also contribute for increasing in fiber diameters. So you stop, you hurt yourself, you stop. You lose some nuclei because mononuclear domain size is maintained. If you look at each individual nucleus, each one has a different transcriptional profile. And so why is that? Why does one cell, which has 1,000 nuclei, why do they each have different transcript profiles? So there's some really cool stuff going on in the mouse world where they can do that. So they actually believe that muscle memory is because some of them are never lost. And so you can, if you hurt yourself and it's a mild injury, and you go back to work within a week or 2, the muscle memory is there. And so you regain your muscle fitness faster.
Do you have any data or have you noticed anything regarding stress and regeneration? So I mean, it's probably not the least stressful situation for a horse to run a derby. So like, have you noticed like maybe that stress extends regeneration, or is that?
So we don't have any information on that kind of stuff. You know, stress comes in multiple formats. So cortisol does go up. Cortisol goes up primarily in an energy metabolism perspective. Is it stressful? Yeah. But, you know, most of the time you think about the horse and the racing component, epinephrine is probably a big thing to play. And not necessarily the detrimental corticoid side. So a delay to repair due to a classic stressor like that probably is an injury. Something that would cause a layup. In a normal situation, we'll have pro-inflammatory cytokine release. And the thought behind that is you have to have macrophages and neutrophils infiltrate in order to take away any kind of damaged material. So it's not really answering your question because I don't know that we can define stress as being a single entity.
No.
Question? Thank you.
Grad students, there's lunch with Dr. Johnson just down the hall. It's free, so get the food.